Okay, tonight we're going to learn a lot of interesting facts about the Marine Biological Laboratory and Woods Hole, or MBL. Our speaker tonight is a leading scientist. He's been associated with the program for their summer program for almost 30 years, and for 10 of those years he was director of their most important program, at least in my opinion, the Neural Sciences and Behavior summer program. So we're going to learn a lot of fun facts about this university that sits quietly in Wood Hole. So I'm going to give you one fun fact that I've learned, which he's not going to be covering tonight, but we're all familiar with the humble fruit fly, right? And many of us know that it's been used in research. Well, one of their scientists, T.H. Morgan, who started at the MBL in 1890, he was on the board, he was a lecturer, he was a researcher. He might not have been the first one to ever use fruit flies, but he did, when he started studying chromosomes and heredity, he used fruit flies because they, there's a one-day generation, so you can see how things change pretty quickly. And his work was so important, he received the Nobel Prize award for that in 1933. And since then, the fruit fly has become like one of the standards tools used to do genetic research, and all that started here on the Cape at the Marine Biological Laboratory. So this is Dr. Zakon. He'll be up here in a minute. He's at the head. Raise your hand, Harold. He and I both live in Falmouth, and we both happen to belong to the Men's Walking Club in Falmouth. So every Friday, a bunch of us get together and we walk down the walking path, usually end up at Woods Hole, and we all meet each other. And I met him one day, and he has fed me at least 100 interesting facts about the MBL. It's just an amazing institution. But you know, he kind of spoon feeds them to me. I was a computer programmer. I don't know that much about biology, so he brings it down to my level. But last year, I had the opportunity to hear him give a lecture to his grad students at the MBL, which is another interesting thing about the MBL. They have all sorts of lectures which are free to the public. So if you're interested in any of them, you can find them and just show up and hear them. And at that point, I realized this guy really knows his stuff, because he went very, very deep. He's an expert on electric fish, for example, and the evolution of the genes within the fish that produce the organ that produces the electric pulse to stun the prey. But his entire lecture was about genes that don't exist in that fish, so that it doesn't get zapped itself when it gets its prey. And it was just a fascinating lecture. I only understood about a third of it, Harold, sorry, but he really knows his stuff. Let me get my cheat sheet here. He spends most of the year at the University of Texas in Austin, where he is, he's also an expert on ion channels. And just his work on ion channels has been referenced in more than 6,000 other research papers. So he spends most of the year as a professor at the University of Texas. He's a former chairman of their section on neurobiology. As I said before, he's the former director of the MBL Neural Systems and Behavior Summer Program. And even though I advertise this as the history of the MBL, he's a true scientist through and through, and he's just going to talk about all the amazing science that has been invented in the past 130 plus years at the Marine Biological Library. And just a little side note before he comes up and starts his presentation. I was in downtown Falmouth this weekend, and there was a man wearing a t-shirt. It said 1888, and it showed a picture of a seahorse, which is the symbol for the MBL. And so I said, do you work at the MBL? And he says, no, I was a student there years and years ago. I said, well, you might know a friend of mine who works at the MBL, Harold Zakon. And all he said was, a very smart man. So I would like to have this very smart man come up and tell you a lot more interesting things about the MBL.
Well, first of all, it's young people at MIT or just in general. So thank you for giving me the opportunity to come. Secondly, let me say, with this men's walking group, it's a group of us, and when I mentioned that I was giving a talk at the MBL, everyone was going, "Oh, when is it? When is it?" And I was saying, "No, no, no, don't come. It's going to be too technical. You shouldn't come. Don't come." The night of the talk, I looked out, there was George. There was George. Is this better?
Yeah.
Yes. Yeah, the only one of the group who came, and I was shocked. "George, what are you doing here?" But true to his word, he came. Okay, now before I start, let me just say a couple of things. First of all, I am a Massachusetts native, even though I'm at the University of Texas. I used to come to the Cape all the time when I was a youngster and in high school, so I have a deep love of this part of the state. I married a Texan, and so I've been at the University of Texas for almost 40 years, and I've been coming to the MBL during the summer since 1995, and our kids grew up here in the summers, so this is home to us. The second thing that I want to say is that I am not an MIT alumnus. However, during 2 years between college and graduate school, I had a job as a research associate at Harvard, and I took a course at MIT, a neuroanatomy course that changed my life by the world's most prominent neuroanatomist at the time. And when I went to graduate school, I went to Cornell, and my advisor was an MIT graduate, and he had gotten his degree with the legendary Jerry Lettvin. So I have sort of an indirect connection to MIT. Well, the reason I wanted to tell you about the MBL is even in Falmouth, the next town over, people will say, "Oh, we've heard about the MBL, but they don't know anything about what has gone on there, its history, or what is currently going on." So I thought I want to tell you about that. So there are some of the buildings of the MBL. And let me give you just a few facts. It's the oldest marine biological laboratory in the US, founded in 1888. It has approximately 250 year-round scientists and staff, but during the summer, it's flooded with visitors from all over the world, all over the US, both scientists who come there to collaborate and interact, as well as many courses in many, many different areas. And scientists come to teach the course, graduate students come to take courses. It's so intellectually vibrant. It's just a wonderful place to be. The MBL has about 100 high school students, I think from all over New England. I'm not really sure of the details, but they have many high school interns, which is a really great thing. The MBL became part of the University of Chicago in 2013. If you want to know details about why it went from a freestanding institute to part of the University of Chicago, I will tell you later. Yeah, it would take the rest of my time. Many species have been studied, and one of my goals today is to tell you why would you want to study animals from the ocean. The final thing is 60, count them, 60 Nobel Prize winners have been affiliated with the MBL. It doesn't mean that they're there year-round on the staff scientists. Many of them came to teach or take summer courses or to collaborate during the summer, but it's a center of intellectual activity. So what I'm going to do is describe 3 different results, 2 of which resulted in Nobel Prizes from the past, and then I'm going to tell you about research currently going on, so you'll see where it's going in the future. So I am a neuroscientist, and the two that I picked are neuroscience related, or two of the three. So one Nobel Prize went to people who studied the giant neuron of squid. And you might say, well, why study squid? Well, it turns out when this work was done in the 1930s and 40s and beyond, you couldn't study neurons, single neurons in us, for example, or a rat or anything. They were too small. But squid have a giant neuron, two, one in each side of the body. And they're big enough you could get electrodes into. And because of their huge size, the first recordings from these neurons were made in Woods Hole at the MBL, and some of the technology used to study them was invented there. And this gave essentially the foundation for our understanding of how neurons work, how they make electrical activity. And that's the field that I work in. So this is very personal for me. Okay, so that's one.
Can you say what the picture is showing us?
Oh, sure. This is an impulse, it's called an action potential, a nerve impulse that lasts 1 millisecond. And Casey Cole, whose name is cut off here, made the first recording of an axon's resistance. Using a Wheatstone bridge, he was able to show that there was a change in resistance in the membrane of the nerve that occurred during the impulse. So during 1 millisecond, there's changes in the resistance of the membranes as ions go in and out. And this is foundational because people had no clue about where the electrical activity came from. You're welcome. Another one, Keffer Hartline studied horseshoe crabs. Well, why horseshoe crabs? Well, as you can see, they have simple eyes. Here is horseshoe crab's eye, higher magnification. You can see the eye, you can see many little individual units, even higher magnification, little cells. Now, the eye of horseshoe crabs and the group that they belong to, crustaceans, insects, that whole large group evolved independently from the vertebrate eye, and yet the principles that they were able, that Hartline and his colleagues were able to deduce from studying how these cells responded to light turned out to be the same principles that during evolution occurred in our own retinas that allow us to process visual information. So, and as you can see here, it's partly a matter of numbers. Simple eyes, they have 1,000 photoreceptors, that is the cells that respond to light. We have 126 million. So by using something simple, they were able to understand fundamental processes of the nervous system. Now, this is out of my area, but Tim Hunt got the Nobel Prize for discovering a key protein, or a bunch of key proteins involved in cell division. Well, he did it with clams. Well, why clams? Well, if you think about trying to study a mammalian egg and sperm and put them together and start understanding how the cells divide from 1 to 2 to 4, etc., it's hard because you can get a limited number of cells at one time. Clams have 10 million eggs and comparable numbers of sperm with each spawn because they have to breed in the water. You know, they don't do it like we do, and I don't have to tell you how we do it. But it's much easier for sperm and egg to meet. But when you're in the ocean and you just throw your eggs out and throw your sperm out, the probability of meeting is very low. So their solution to this is to make tons of eggs.
So Hunt could take a lot of sperm, a lot of eggs, from another clan, get the sperm, put them together, and all these millions of eggs would begin dividing at the same time. So if you're looking for proteins that are involved, it's synchronized to the dividing of the eggs. One to, each egg to 2, each of those to 4, and these are going on in unison in all the millions of eggs. So he discovered a protein that appears just before the cells divide, and then it disappears as it's broken down. And with the next cell division, another protein appears, and those, when he looked, are the same as us. They're in the same as other organisms. And so this gave a clue to important proteins that we now know are involved in, mutations can cause embryonic disorders or cancer. So he didn't go in to study cancer. He went in to just look at what are the proteins involved in cell division, but it turned out to be a very important result. Now this isn't a Nobel Prize, but it will, there's another prize I'll tell you about that these guys were awarded, but Fred Bang, Jack Levin were studying horseshoe crabs. And what they found was there are cells that horseshoe crabs have in their blood that are extremely sensitive to bacterial toxins. And if these cells encounter a bacterial toxin, there are all these granules inside the cells, and they will release a substance that starts clotting the blood to try and trap the toxins. And it turns out that there is nothing that we know of that is as sensitive to bacterial toxins as horseshoe crab blood. And one other thing, just for your interest, yes, horseshoe crab blood is blue. They're real blue bloods. And the reason is because we use iron as the component of hemoglobin that binds oxygen. They use copper. So it's just a different element, but it causes the blood to be blue. Now, what I want to do, you know that a picture's worth 1,000 words, but a video is worth 1,000 pictures. So I'm going to see, if I can show you a video. Now, this is about the Golden Goose Award, which is an award given for, the goose that laid the golden egg. These are awards given to people who have discovered, really important things. This is good video. Can you all hear this?
The whole village of Woods Hole is really oriented towards science, and we grew up as kids of scientists.
I don't remember the first time I saw a horseshoe crab, but I do know that I called them limulus before I called them horseshoe crabs. That's living in a scientific family. My father, he was very enthusiastic about science, and he was known as an educator as much as he was known as a researcher.
I was studying platelets, and it was early in that period that Dr. Frederick Bang approached my chief and related to him the work he had done, in which he demonstrated that bacteria could cause blood coagulation in the horseshoe crab, and suggested that if hematologists joined him at Woods Hole, some interesting results could be obtained. And so in the summer of 1963, I went to the Marine Biological Lab at Woods Hole to join Dr. Bang's laboratory. I have to emphasize that I had never seen a horseshoe crab in my life. Dr. Bang, without any additional explanation, told me to put my hand in the tank and grab a crab. Our goal was to draw comparisons between the human blood platelet and the blood cell of the horseshoe crab. But when I started to handle the blood of limulus, it clotted very rapidly, and I was absolutely stumped and stuck. realized that I had a very powerful and practical mechanism for detection of bacterial endotoxin.
Dan understood exactly what the importance of it was, but I don't think the general scientific community understood the importance of it for a long time.
Slowly, the test became accepted to be reliable and specific for endotoxin. The impact of the Limulus test and its ultimate major use in the pharmaceutical industry has been extraordinary and beyond anything I ever considered. It is the gold standard for endotoxin detection throughout the world.
The ability to measure in a precise way the amount of endotoxin in things we are exposed to every day has a societal benefit that I think few people recognize. We are where we're at today in large part because of their good work.
I think that my work is an excellent example of the importance of conducting and supporting basic biological research, even when there's no obvious practical benefit at the time the studies are started. The history of investigative science indicates that early failure is a common feature of many discoveries which turned out to be very important.
And that's an important point. We often, those of us who do basic research, we don't necessarily know what the application will be. We just look for understanding the world around us, and the things you might never imagine that it can be applied may be applied.
Closer to the mic, please.
Excuse me?
The mic up toward you.
Sorry.
We want to hear you.
Okay. So that was just a few highlights of the amazing amount of work. But rather than dwell on the past, let's talk about the present and the future. So I'm going to give you a few highlights of work going on now. So I will just say there's a big program looking at cephalopods. Those are squid and octopus, and there are many things about them. For one thing, they can camouflage themselves in an amazing way. They can control, they have cells all over their bodies with pigments, and their brain can control each cell and cause it to expand or contract. They can change the pattern on their bodies to match a chessboard or to match anything you put them on. And they can raise the skin with little muscles to change their texture. So scientists at the MBL are working with the military in terms of camouflage. They have cognitive abilities. You know, we have evolved a brain, but they did too. If you compare a squid with a clam, which is its relatively close relative, it's not a cephalopod, but they're all mollusks. Think about how big the brain of an octopus is compared to a little clam brain. And so it's a way we can understand how does intelligence evolve, because if you only study it in vertebrates like us, you only see that it evolved one way. Did octopi evolve intelligence in a different way or the same way we did? We don't know, and that's why it's understudied. Vision, they have great eyesight, other interesting senses. I will say something briefly about RNA editing in a minute, but let me play this video.
Complex behaviors.
Wait a minute.
Cephalopods captivate the imagination, at once appearing both familiar and alien because of their complex behaviors. For millennia, they fought for dominance in the oceans, competing with fish and other vertebrates. Over time, they have developed the ability to control their skin's tones and textures to produce near-perfect camouflage. Their eyes and ours share a common design, allowing them to better capture prey and avoid predators. Cephalopods have many hidden novelties, like their ability to edit their genetic information on the fly. At the Marine Biological Laboratory, we are combining state-of-the-art cephalopod culture with cutting-edge genetics to explore the biology of these fascinating organisms.
I'll just say one thing very briefly. So, you know, our genetic information is encoded in DNA, and DNA then makes something called RNA, which is taking the information from the genome, and then that goes in our cells to make proteins. So it turns out that, I'm sure you know, mutations in the DNA can be copied in the RNA and copied in the protein and make a defective protein. And that can cause many kinds of diseases. I've listed just 3. Sickle cell anemia, cystic fibrosis, Alzheimer's disease. It's a long list. So why am I telling you about cephalopods? Well, RNA editing, let me just go back. One can change the RNA through a chemical process that occurs in all of us. We do it too. But it turns out, of all the animals known, cephalopods have the most RNA editing. They change the RNA of many different, for many different proteins. So by studying RNA editing in cephalopods, we're learning a lot about using this process of RNA editing as a therapeutic tool. Now, I'm going to finish with one last comment and video. The Eugene Bell Center for Regenerative Biology and Tissue Engineering at the MBL was named after Eugene Bell from MIT. He is considered the father of regenerative medicine. He was first able to take human cells for skin, embed them in a matrix where they would essentially form skin, both dermis and epidermis, all the parts to skin, and use them to as replacement skin for, say, burn victims. But it was using the patient's own cells, so there was no immune rejection. And so his work, he was a founder of, regenerative medicine, growing tissues in culture to use to replace cells. So this was named after him. And as you can see, the Bell Center does a lot of things, but they study stem cells, which are the cells that we have that are uncommitted as to their identity, but can be pushed to become a skin cell, or a brain cell, or a liver cell, depending on what is needed. And they study regeneration in axolotls, which are a kind of salamander, sea stars, that's starfish, lampreys, which are a kind of fish, that's the mouth of a lamprey, and nematostella, which is a sea anemone. But the point is, there are some of these organisms you can take, cut them up into pieces, and they regenerate a complete body. It would be like cutting off your hand, putting it down, and from your hand, the whole rest of you comes out. So how they do this, and how different organisms can regenerate parts and regenerate nervous systems. For example, some salamanders, you take out the retina, they regenerate a whole new eye. You cut off a limb, they regenerate a new limb. So this is why they're studying these organisms. And I'm going to finish with one final video. Jennifer Morgan, who is the director of the Bell Center, can tell you this better than I can about some of the work they're doing.
Morgan, and I am an associate scientist at the MBL. I'm also the director of the Eugene Bell Center for Regenerative Biology and Tissue Engineering. The Bell Center for Regenerative Biology and Tissue Engineering was founded here at the MBL in 2010 through the very generous gift of Millicent Bell in honor of her husband, Eugene Bell, who was an absolute pioneer in the field of tissue engineering. A couple of things that we're really interested in are understanding what are the mechanisms that support successful regeneration of the central nervous system, which is your brain and spinal cord. And we're also really interested in understanding how diseases of the nervous system like Parkinson's disease and Alzheimer's disease affect how the synapses in your brain work. Our main model organism is the lamprey, which is looks like an eel, but is a fish. And the lamprey is really cool because it's a very primitive vertebrate. It evolved about 550 million years ago from a common chordate ancestor. And even though they're so primitive and so old in evolutionary terms, it turns out they have brain and spinal cord structures that are very similar to the ones that we have in our own bodies. Unlike humans, if you sever the spinal cord of a lamprey without any treatment or intervention whatsoever, you can put the animals back in the tank, and over the time course of about 3 months, they're able to fully recover their ability to swim. So the real question is, if they can do it, why can't we? Because the similar injury in humans, the person is going to be confined to a wheelchair or have other sensory and motor problems likely for the rest of their lives. So this big group of collaborators came from 4 or 5 different institutions over several summers here at the MBL to start to look at that molecular recipe for successful regeneration. The remarkable thing that we found was that lampreys are turning on genes that promote growth in the peripheral nervous system of humans. That tells us that there's an evolutionarily conserved program that's able to drive nerve regeneration from this very primitive lamprey all the way to humans. And if that's the case, if we could figure out how to turn on that program in humans, in the spinal cord or in the brain, we might have better successes in the clinic. Thomas Hunt Morgan was one of the pioneers in the field of regeneration. He did a number of studies, many of which happened here at the MBL. Having the current work from the Bell Center for Regenerative Biology happening now is really kind of a way to have this long-term legacy at the MBL in a place that's been a driver of the field.
That's all I have to say. Questions, anyone? Thank you. Yes.
Just 1 second, we have a microphone, so you'll be able
Oh, you want to use this microphone? Oh, I see. Got it.
Tom, thank you. That was extraordinarily interesting, and I thought it was a great selection of the past and looking forward, and I thank you for that. It takes a lot to bring together a team and create an environment, and the history of being founded in 1888 and being so successful is probably the legacy of lots of people, lots of effort over many years, just showing, well, how do you maintain your funding? I know from a career in academic medicine, both the benefits of training in excellence centers, but I know the challenges of ongoing funding. Highly competitive field, there's never enough money from NIH and other federal sources. There's great money from the private sector, but then again, that's all focused towards applications. And it's a very competitive field, and in order to be successful, you have to write grants, you have to get funding. So could you give us some insight as to not only the intellectual abilities, but the administrative and financial mechanisms that might keep you going, or what challenges you find?
Now you're getting some insight into why the MBL was taken by the University of Chicago.
I thought so, as somebody who's been on the faculty at University of Chicago, among other places.
Well, actually, so it is a difficult thing. It's difficult to get funding in established institutions, and for most of its history, the MBL was just a group of people who came together, a group of scientists who came together, and the director was somebody they elected, and everything was catch as catch can. People, brought stuff, or they had grants that they could use. And one problem is, as I'm sure you know, science gets bigger and bigger, and the things you need to do cutting edge science increase. So there's been a lot of pressure everywhere, not just the MBL, to keep the dollars coming in so you can do the work. Just part of this was, in 2013, the University of Chicago came in because there was a deficit, and essentially said, we'll, take over, we'll buy the MBL and fund you, give you some extra funds to keep you going. And that's been a formula for about 10 years. Now, you might say, why the University of Chicago?
Why'd they do it?
Yeah, well, it turns out the first director and early directors of the MBL were from the University of Chicago. And there was actually a very old connection since I guess it would be 1890s, when it went from just being classes that people held and summer research in 1888. After a few buildings were built, and there was an entity for someone to be the director of, people from University of Chicago came, and so there's a very long-standing collaboration or connection to the University of Chicago.
Did Chicago already have funding then? Were they in turn looking for a laboratory that was performing, and they already had some funds to start with?
Well, did everyone hear that?
No.
Maybe, yeah, he was asking, well,
Is that Chicago, did they already have some funding for this kind of research and just discovered that, gee, some of it's going on down here, Woods Hole?
Not so much that. They've been many of their investigators were involved in research going on at the MBL. So there's a long history. It was really more that there was a kind of an opportunity where the University of Chicago had funding and realized the MBL was needing a little more to keep up, that they decided to do this.
Good.
Any other questions?
This may not be squarely on your topic, but one thing that I wondered, you're talking about the limulinus of the horseshoe crab. Are they endangered now? I mean, if they're being harvested, and is anybody farming horseshoe crabs?
Yeah, good question. It's not my area, so I don't know details, but I know that they are being taken for their blood, but they can be bled and returned to the environment.
Oh.
But I will tell you, last year or the year before, my wife and I were walking around some of the docks, and we came upon a pile of dead horseshoe crabs, or dying horseshoe crabs. They were just lying on the dock, and some still moving a little, and many of them were dead. And we thought, what is going on? So we made a few phone calls to find out, and it turns out that they are being used by for bait. And there's a certain number that I think commercial fishermen can take. So for those reasons, and for reasons of loss of habitat, and many other things, they are being severely impacted. But it's not the pharmaceutical industry that's doing it primarily. Yeah. Yeah. George?
Excuse me, what?
What eats horseshoe crabs? I mean, what are they trying to catch?
Oh, I don't know exactly, but I think it's like pieces. You know, chop them up into pieces and bait a hook. But, you know, I will say this about horseshoe crabs. Like anything, you know, you focus on one organism and you look at its life, and if you don't look at the ecology around it, all the other things that eat it, or that it eats, or are impacted, you'll have a very small, narrow view. So horseshoe crabs, not intentionally, but they provide lots of eggs for migrant birds, because seabirds will eat the eggs. And so, you know, for any organism, you can trace out a network, a web of all the other organisms they interact with. So, yeah, we have to be careful.
I can speak loudly enough. Question of ignorance. How is the MBL related to Woodsville Oceanographic Institute? Is it
Good.
contiguous? Is it a subset?
Yeah. Very good question. Hui is a copycat. In 1930, it was decided that the U.S. needed an oceanographic institution. There's a distinction. So the MBL is mostly working with, where it does work with marine organisms inshore, fairly close to the shore, and the MBL has a boat that goes out to catch squid, for example. But an oceanographic institution gets way out and way deep. And so in order to establish that capacity, Hui was set up along the lines of the MBL in 1930. It's obviously been very successful. I'm sure you know about the Hui MIT program, so they have a close connection with MIT. And they cooperate, but they're very separate.
Thank you. How close are they to actually helping people who either have brain damage or nerve damage to actually reproduce it?
Well, for a lot of these things, we're a long way off in terms of what you can do in the clinics. But the amount of information that's being gathered is just getting, you know, greater and greater. I mean, I will tell you one or two things that, not necessarily with the MBL, but for example, Huntington's chorea, which is a motor disease, people have discovered the genes now for it. Well, this is about 20 years ago. So we're understanding more about genes and the proteins that they make and how they interact. And the big problem, even once we understand what causes a disease, is getting therapies that are useful, that can be used, because you can't, it gets too complicated, but things are difficult to actually bring to the clinic. But more and more, it's getting closer and closer to real application for some of these things. Well, no, go ahead.
Anybody else?
Well, thank you very much.
MIT Club stainless wine glasses.
Oh, fantastic. Thank you very much.